I'm Alison Singer with the Autism Science Foundation, and I'm here now with Dr. Joseph Buxbaum of the Seaver Autism Center at the Mount Sinai School of Medicine. Thanks so much for joining us.
It's a pleasure.
You presented a study today that showed a new treatment that may be efficient in reducing some of the learning deficits in the SHANK3 mouse model. Can you tell us a little bit about your study?
Sure. So one of the big breakthroughs in autism today is using genetics to find genes to make mice that have the same mutation you find in autism, and then study the mice, and then find novel targets for drug development. So SHANK3, as I said, accounts for about 1% of autism, and we've made the mouse that has the SHANK3 mutation, and we're able to determine what goes wrong in brain development when you mutate SHANK3. And so what we found is certain forms of normal adaptation of nerve cells don't work so well, and we found a drug that seems to reverse it in the mice.
Now, this is similar to what we've seen in the Fragile X mouse. Is this the first time that there's a targeted therapeutic for a non-Fragile X, non-Rett mouse model?
Yes. So this is the concept is similar in that you make a mouse model and then you study the biology of the mouse. But what's going wrong in Fragile X, for example, is very different than what we see here. So this is a different class of autism, if you will. And here, too, we're making progress in trying to develop novel therapeutics.
And how long does it take once you've discovered a novel compound that's effective in a mouse? How long does it take until we go through human clinical trials and have something that may be useful in people?
Well, it certainly takes years, but it also depends on what compound you're using and what's available already, what's in a pipeline for already in a pharmaceutical company, and what needs to be developed from absolute scratch. With the compounds that people are using, they're trying to use compounds where there's already an existing compound, so they can shorten that process to, a few years rather than more than that. But it's a case-by-case phenomenon.
A lot of parents question the whole concept of a mouse with autism. Can you talk a little bit about how you look at the mice and how you phenotype the mice and how you actually make a mice that shows characteristics of autism?
So, of course, mice don't get autism, but mice have a very similar brain structure, brain development, like humans. And so you can ask, for example, how is the brain development changing? You can ask how nerve cells communicate with one another in the brain. You can even do behavioral studies. So, for example, mice are very social animals, and some of the mice where we introduce a mutation that you find in autism become much less social. So that becomes a good endpoint. If you were to try a drug, you'd say, do I reverse this social deficit? Do I reverse the deficit in communication between nerve cells? So these are all, they capture facets of autism, but much more importantly, they are endpoints to assess how well your drug is working.
And this is a model that we've seen used in other diseases and disorders. Is that correct? The gene discovery to animal model?
Absolutely. So there are many examples of mouse models for either other brain disorders or for even disorders not in the brain. Certainly, people have talked for years about the Alzheimer mouse models, where they introduce some of the genes that we find in Alzheimer's disease, and those have all been used for therapeutic trials. I think generally this approach of identifying the cause of a disease to then develop targeted therapies is really where all of medicine is going. It's been called personalized medicine or individualized medicine, and it says understanding the cause will lead to specific treatments that may need to be tailored to each person or each group of people individually.
So what are the next steps for you and the members of your lab?
So we are continuing to look at these mice very intensively. We are trying to use an already existing version of this drug that's already approved for use in children in the mice to see if that also works.
And what is the drug?
Insulin-like growth factor 1, also called IGF-1. But we also have found in the same studies that some receptors for which there's already a very large armamentarium, if you want to use that word, a very large number of compounds that target this particular receptor. We also found that those receptors are altered in these mice, and so we have other opportunities to try additional therapies in the mice. And our hope is that over the course of very few short years, we'll have a list of compounds that we think are working in the mice that we, from other studies other people are doing, we know are safe for humans, and then we can consider clinical trials. Whether it'll be my lab or other labs in the field, I can't say, but certainly that's the commitment of the researchers to bring us to clinical trials as soon as possible.
Thank you so much for joining us today.
You're welcome.